Hello friends, welcome to entranceadvisor.com. In today's video, I'm going to talk about the molecular basis of inheritance, class 12 biology, that is unit 7, and this one is lecture 3, the last part. So let's start the video. If you are new to this channel, please hit the bell icon and do the subscribe button. So let's start. What is genetic code? Genetic code is the relationship of amino acid sequence in a polypeptide and nucleotide base sequence in mRNA or the antisense strand of the DNA. Crick in 1961 found that the deletion or addition of 3 consecutive nitrogen bases caused less disturbance than 1 or 2 bases in the DNA or the T4 bacteriophages. So, Nirenberg and Mathy in 1961 prepared the polymers of the individual nucleotides and found polyuridylic acid. The poly-U stimulated peptide sequence, that is polyphenylalanine, poly-A polylysine and poly-C polyproline. Poly-G did not function as it formed triple-stranded structure. They further found that the number of the amino acids in a peptide was a 1/3 of the number of nucleotides in the polymer. Trna in 1964 observed that the nucleotide copolymer UGUGUGUG stimulated peptide with the alternate cysteine and valine. Okay. So for this work, the Trna Hawley was awarded the Nobel Prize in 1958. Then Ochoa in 1965 employed other copolymers and therefore cracked a number of codons. The DNA and the mRNA codons are complementary. AAA of the DNA becomes UUU on mRNA. That's very simple. So, genetic code is the relationship of amino acid sequence in a polypeptide and the nucleotide base sequence in the mRNA. Okay. It directs the sequence of the amino acids during the synthesis of proteins. George Gamow suggested that the genetic code should be a combination of 3 nucleotides to codon 20 amino acids. A.T. Trna developed the chemical method for synthesizing RNA molecules with defined combination of bases. Marshall Nirenberg's cell-free system of protein synthesis finally helped the code to be deciphered. So the genetic code, the sequence of nucleotides, is the DNA and the RNA that determines the amino acid and the sequence of proteins. Make it very clear in your mind. And though the linear sequence of the nucleotides in the DNA contains the information of the protein sequences, proteins are not made directly from the DNA. And instead, a messenger RNA, mRNA molecule is synthesized from the DNA and directs the formation of the protein. The RNA is composed of 4 nucleotides, adenine, guanine, cytosine, and uracil, and the 3 adjacent nucleotides constitute a unit known as the codon, okay, which codes for an amino acid. For example, the sequence AUG is a codon that specifies the amino acid methionine and start codon. And there are 64 possible codons, 3 of which do not code for amino acids but indicate the end of the protein. The remaining 61 codons specify the 20 amino acids that make up proteins, and the AUG codon, in addition to coding for methionine, is found at the beginning of every mRNA. As I said, it's the start codon and indicates the start of a protein. The methionine and tryptophan are the only 2 amino acids that are coded for by just a single codon, AUG and UGG respectively. Okay. The other 18 amino acids are coded for by 2 to 6 codons, because most of the 20 amino acids are coded for and by more than 1 codon. That is called degenerate. I will be coming to this later. What is degenerate? So the genetic code, once thought to be identical in all forms of life, has been found to differ slightly in the certain organisms and in the mitochondria of the same eukaryotes. And nevertheless, these differences are rare, and the genetic code is identical in almost all the species. With the same codon specifying the same amino acids, the deciphering of the genetic code was accomplished by the American biochemist Marshall W. Nirenberg, Hawley and Herb Trna in 1960s, as I said, and the nucleotide triplets specifying different amino acids are shown in the table. Just have a look in this table very carefully. Okay, this is the RNA codon table and the amino acids are listed below. This is UCGAG and the permutation and combination resulted in this. Okay, and these are the stop codon. You have to remember the name of the stop codon and the start codons for your exam purpose. So the salient features of the genetic code are the codon is a triplet. 61 codon code for amino acids and 3 codons do not code for any amino acid called the stop codon. That is the UAG, UGA and UAA. So it is a triplet code. That means 3 adjacent nitrogen bases constitute a codon and specify one amino acid. Got it? The codon is unambiguous and specific and code for one amino acid. Okay, this means the codon is commaless. There are no pauses so that the genetic code reads continuously. Any change in the nucleotide sequence will make the code read differently. The possible number of triplets is 64. Okay, T to the power 3, but the amino acids involved there are only 20. Find it. The codon is degenerate. Some amino acids are coded by more than one codon. Got it? What does degenerate mean? And the codon is read in the mRNA is a contiguous fashion without any punctuation. I said it is commaless. The codon is nearly universal and AUG has a dual functions. It codes for methionine and also acts as initiator codon, start codon. So they are non-overlapping. Okay, the possible triplets of the number of triplets that is T to the power 3, but the amino acids involved there are 20. I said the non-overlapping means adjacent codons are independent with no base being member of the two codons. Universality, it's their universal, and the codon specifies the same amino acid in all organisms from virus to human being. The genetic code remains same. Okay, they are non-ambiguous in nature. I said, or they are nearly universal. They are not totally universal. There is various controversies regarding this till now, and the codon specifies the amino acid in all organisms from virus to human beings to bacteria related any other species. Okay, the one codon specifies only one amino acid and not any other. Remember this. So I said they are degenerate. This is very important characteristics. You have to write this in your exam whenever asked. And the tryptophan is specified by the UGG and methionine, but only AUG. All other amino acids are specified by 2 to 6 codons. Example, phenylalanine by 2 UUC and UUU. Okay, and the arginine by the 6, as you can see in this chart, AGA, AGG, GCA, CGC, CGG and CGU. Most probably you heard about the term cistrons. What are that? The expressible parts of the genetic information contained in the DNA occurs in a linear sequence called the cistrons. You get to know and obviously get to learn about this in your higher studies, or most probably. Each cistron has a fixed polarity with a start point and an end point. They are collinear. The sequence of the codons of DNA or mRNA correspond to the sequence of the amino acids in a polypeptide. So there is a start signal. The first codon starting in the polynucleotide is AUG, methionine specific, but in the prokaryotes it can be GUG and even it's UUUG. Okay, the leaving. In all the cases, they specify the methionine as the first amino acid, though GUG and UUG specify different amino acids inside the polypeptide chain. GUG valine and UUG leucine. That is the stop signal, and the start. Obviously, there is a start and obviously there is a stop. I said the stop signal termination of the polypeptide signal or the synthesis occurs one of these nonsense or the termination codons. That is the UAA, UAG and UGA. This is UAA called the ochre and UAG called the amber and UGA called the opal. Okay, remember the related codons of the amino acids with the similar properties have usually related codons, and aromatic amino acids such as tryptophan, UGG, phenylalanine, UUC, and UUU are the tyrosine, UAC, and UAU. There is another hypothesis known as oval hypothesis that is very important. That is the anticodon, the 5' end base of the tRNA anticodon is able to oval and get paired with even non-complementary base of the mRNA. Therefore, the first two bases of the codon are more important in determining the amino acid, while the third one may differ without affecting the codon or the coding verbal loose pairing. CCA and CCC, CU and CCU all specify amino acid called a proline. So what are mutations? Let's come on to this part. A change of a single pair or the single base pair point mutation in the sixth position of the beta globin chain of the hemoglobin results due to the change of the amino acid residue to glutamate to valine. This results into disease condition known as sickle cell anemia. This one is sickle cell anemia. And the mutation is any change or any deformity caused or any sort of differences caused by base pairing changes or something like that is called mutation. So insertion and deletion of the three or the multiple bases insert the different delete or one or more multiple codons. Hence, one or more amino acids are reading frame remain unaltered from that point onwards. Such mutations are called the frame shift insertion or the deletion mutations. There are various types of mutations. Now you don't have to remember all this. This is deletion, there is duplication, there is inversion, there is insertion, and there is translocation. There are multiple types. You just know the name. Okay. Now for Nauvin and for the change from glutamate to valine, the sickle cell anemia occurs. So the central dogma describes how the DNA makes its own copies by the DNA replication, then codes for the RNA by means of a transcription, and RNA codes for the protein by the translation. In this lecture, I'll be mainly discussing about the translation part. For the creation of the new generation, the genetic information stored in the DNA has to be transferred from one generation to another. The process begins with the replication of the DNA followed by the transcription and translation. Let us have a discussion of the translation with the protein synthesis and the structure of the tRNA. The tRNA, the adapter molecule. The tRNA called as the adapter molecule. It has an anticodon loop that has bases complementary to code present on the mRNA and also has an amino acid acceptor to which amino acid binds. The tRNA is specific for each amino acid. The secondary structure of the tRNA is depicted as a clover leaf-like structure, and in actual structure the tRNA is a compact molecule which looks like the inverted L. Okay. So the central dogma of the molecular biology proposes a unidirectional or one-way flow of the transformation from the DNA to RNA, transcription from RNA to the polypeptide translation. Commoner of the circular flow information say does that DNA to RNA, protein to RNA, and RNA to DNA. Its cycle basically the transcription or the RNA synthesis occur over DNA, and the translation or the protein synthesis occurs over ribosomes, and the two are separated in time and space. This protects the DNA from respiratory enzymes and RNAs from the nucleases. So as the DNA contains all the hereditary information, it is called a master copy of the genetic information. It replicates or forms its carbon copies when the new master copies are required. Normally, it forms working copies in the form of mRNA. As you can see, here is a detailed structure of the tRNA or the clover leaf-like structure. The DNA is transcribed into the mRNA on the basis of their complementarity. The translation is the process of the conversion of nucleic acid information into amino acids. There is no complementary between amino acids and the mRNA. Hence, the translation is not controlled by the complementarity but by the genetic code. Since the amino acid cannot read this genetic code, they are dependent on an adapter molecule in sequence, and this adapter molecule is called the tRNA. You know, you need an adapter to transfer the copies from one device such as laptop or to the phone. There is an adapter, okay? So for the dependence, you know, you tend to depend on something. So here the tRNA also transfers. This is a thing which transfers the RNA. That's why they're called the transfer RNA. The codes, the codes embedded in the RNA that are transferred to make a protein. Got it? So the protein synthesis or the biosynthesis of proteins is known as the translation. The protein synthesis occurs over ribosomes, which are also called the protein factories. Okay? The ribosomes may form a rosette or helical groups of 5 to 20 called the polyribosomes. The ribosomes of a polyribosome are held together by a strand of mRNA. The distance between two ribosomes of a polyribosome is generally 34 nanometer. Each ribosome has two parts, smaller unit. You know, the ribosomes has two parts, one is smaller unit and one is larger unit. The smaller unit is 40S subunit and the larger one is 60S subunit. In case of prokaryotes, the smaller is 30S, and in case of prokaryotes, the larger is 50S. And the large 50S subunits of prokaryotes has 20S and 5S subunits again. Okay. So there are different types. You may not remember this. And a tunnel is found between the two for the mRNA. The larger subunit that has a group for the protection and the passage of the polypeptide. There, the function remains. The site A is called amino acyl tRNA site, enzyme polypeptidal transferase site, and the binding site for tRNA. And the smaller subunit has a for attachment site mRNA. Okay. Alongside larger subunit, each forms the P site of the peptidal transfer or the donor. They are the binding sites for the initiator factors, elongation factors, translocase, GTPase, etc. The raw materials for the protein synthesis are the amino acids, as you know very well, and mRNA, tRNAs, and the amino acyl tRNA synthesis. Number 1. The amino acids. Some 20 types of amino acids are there, and which constitute the building blocks of the monomers of the proteins, and they occur in the cellular pool. And second one is mRNA. It carries the codon or the coded information for the synthesis of one unicistronic for more than polypeptide polycistronic. It codes codons are recognized by the tRNAs. The tRNAs lastly, and they are meant for carrying the amino acids over the mRNA strand according to the codons of the latter. One tRNA picks up a single amino acid. The process of attachment of tRNA with a specific amino acid is called charging. However, one amino acid can be specified by 2 to 6 tRNAs. The amino acid tRNA synthesis are the enzymes that are specific for the particular amino acids and their tRNA. So let's move on to this process. The translation is a process of the polymerization of amino acids to form a polypeptide. The order and the sequence of the amino acids are defined by the sequence of bases in the mRNA. The amino acids are joined by the polypeptide bonds. It involves the following two steps. That is the charging of the tRNA, and second one is the formation of the peptide bonds between the two charged tRNA. The start codon is AUG, and mRNA has some additional sequence that are not translated called the untranslated region or the UTR. For the initiation, the ribosome binds to the mRNA as a start codon. The ribosome moves from the codon to codon along the mRNA for the elongation of the protein chain. As the end, release factors to the stop codon, terminating the translation and release of the polypeptide from the ribosome. So, the process by which the mRNA codes for a particular protein known as translation. In the process, the ribosome translates the mRNA produced from DNA to chain of specific amino acids. This chain of the amino acids leads to the protein synthesis. It is a process where the expense of the ATP is required and this energy is given by the charged tRNA. The whole machinery of the translation is present in the ribosomes. And the ribosomes consist of a bigger subunit and a smaller. I said about this, the larger subunit in turn consists of two molecules tRNA that are placed close to the peptide bond could be developed at the expense of the sufficient energy. The mRNA enters a smaller subunit which is held by the molecules of the tRNA and the complementary codon that exists in a bigger subunit. Hence, the two codons are held by the two molecules of the tRNA placed near each other and the peptide bond is produced among them. When the process repeats, a charge chain of the amino acid is synthesized and the stop codon ribosome releases the amino acid chain. So I'm going to discuss about the charging and all the process involved in this. Okay, let's start. Charging of means the activation of the amino acids. It occurs in the two steps. In the first step, the amino acid combines with the specific amino acid tRNA. Okay. Synthase enzyme, that is in the presence of the ATP in the form of an amino acid adenylate enzyme complex, and the pyrophosphate is released, as you can see. Okay, amino acid present in the complex is activated amino acid. In the second step, the charging and activated tRNA amino acid attaches to the CCA or 3' end of the specific tRNA to form the amino acid or AA tRNA charged tRNA adapter molecule. Second is initiation. It requires the GTP and Mg2+. And proteinaceous initiation factors. Prokaryotes have 3 initiation factors and eukaryotes have 9 initiation factors. These are not required for you now, for now being. Remember the initiation, which tends to initiate and produces an initiation complex to carry forward the process, and then the elongation occurs. As I said in the previous slide, a new amino acid tRNA comes and lie over a site codon by means of GTP and elongation factor, carries forward. Polypeptide bond is established between the two carboxyl, and as you know, there in the protein there is two groups, amino acid and nitrogen. There is two ammonium group and P site, and the amino group at the A site with the help of the enzyme peptidal transferase synthase. Okay, the latter in the RNA enzyme probably the larger subunit of the ribosome. Connection between the tRNA and amino acid of the P site breaks and the A site tRNA comes to be at the dipeptide. So their freed tRNA of the P site moves to the E site and then slips away. By means of translocase enzyme, the ribosome moves in relation to the mRNA and the polypeptidal carrying the tRNA comes to lie on the P site and a new codon is exposed at the A site. Okay. By this process, you can see in this growing polypeptide chain incoming tRNA bound amino acid. This is a very simple or schematic diagram for you to understand only. Then the termination occurs when they receive the termination codon, and after that modification of the protein occurs. You will be knowing more about this process deeply in the higher classes. So how the regulation of the gene expression takes place? So you need to know that all the genes are not needed constantly. The genes needed only sometimes called the regulatory genes and that are made to function only when required and remain non-functional at other times. Such regulated genes are called therefore required to be switched on or off when a particular function is beginning to stop. The Lac Operon. The Lac Operon consists of one regulatory gene and that is I, the 3 regulatory genes YZA. Gene I codes for the repressor of the Lac Operon and Z gene codes for beta-galactosidase that is responsible for the hydrolysis of disaccharide and the lactose into monomeric units, galactose and glucose. And gene Y code for the permease, which increases the permeability of the cell. The gene A encodes for transacetylase. So what is an operon? Operons are the segments of the genetic material which function as regulated units or units which can be switched on and switched off. Operon concept was given by 2 French scientists working at the Pasteur unit, Francis Jacob and Jacques Monod. They are the two and they were studying lactose utilization in the mutants of Escherichia coli, that is E. coli. And operon consists of 1 to several structural genes, 3 in the Lac Operon and 5 in transcript tryptophan operon of the E. coli. Okay, and they are a promoter gene, a regulator gene, repressor, an inducer or co-repressor. The operons are of two types, inducible and repressible. Hope you understand. So lactose is a substrate of the enzyme beta-galactosidase and it regulates switching on and off of the regulatant, so it's called an inducer. Regulation repressor is referred to as negative regulation by the Lac Operon, and operation of the Lac Operon is also under the control of the positive regulation only. So here is a diagram. So the Lac Operon is, the T is missing. The Lac Operon is an operon or the group of the genes with a single promoter transcribed in a single mRNA. The genes in the operon encode the proteins that allow the bacteria to use the lactose as an energy source. So what happens? In absence of inducer, the repressor binds to the operator region and prevents the RNA polymerase from transcribing the operon. So the repressor binds and the translation cannot occur. In the presence of inducer, the repressor mRNA, there is inactive repressor is formed. So there is no blockage and the Lac mRNA is transcription occurs, and then obviously the translation occurs and get results in the beta-galactosidase permease and the transvacillates the different proteins. Let's see this in more detail. The Lac Operon of the E. coli contains genes involved in the lactose metabolism. It's expressed only when the lactose is present and glucose is absent. Two regulators turn the Lac Operon on and off in response to the lactose and the glucose levels, the Lac repressor and the catabolite repressor or the CAP. The Lac repressor acts as a lactose sensor. It normally blocks the transcription of the operon but stops acting as a repressor when lactose is present. The Lac repressor senses lactose indirectly, though its isomer allolactose, and catabolite activator protein CAP acts as a glucose sensor. It activates the transcription of the operon, but only when the glucose levels are low. The CAP senses glucose indirectly through the hunger signal molecule of the CAMP. So here is the structure of the Lac Operon, more details. Their site is promoter and there is RNA polymerase. When operator is blocked by the repressor, it blocks RNA polymerase to transcribe. So and this is the CAP site that promotes the RNA polymerase binding too. When there is no lactose, when the lactose is not there or absent, the Lac repressor binds tightly to the operator and it gets in the RNA polymerase way, preventing the transcription. Okay. With the lactose, allolactose rearranged lactose binds to the Lac repressor and makes it let go to the operator, and the RNA polymerase can now transcribe the operon. Okay. When there is low glucose levels, when the low glucose levels are present, CMP is produced. The CMP attaches to the CAP, allowing to the bind DNA, and CAP helps the RNA polymerase bind to the promoter, resulting in high levels of transcription. And when there is high glucose, when the glucose levels are high, no CMP is made, so CAP cannot bind to the DNA without, and so the transcription occurs only at a low level. So now it's time for differentiation and development. And the goal of HGP, what is Human Genome Project? It was launched in 1990 to find out the complete DNA sequence of the human genome using the genetic engineering technique, bioinformatics, to isolate and clone the DNA segment for determining the DNA sequence. So what is the goal of that HGP? Identify all the genes in the human DNA. They determined the sequence of the 3 billion chemical base pairs that make up the human DNA. Store this information in database, improve the tools for data analysis, transfer the human information to the other sectors to address the legal, ethical, and social issues that may arise due to the project. The project was coordinated by the US Department of Energy and the NIH, and the method involved the two major processes. The first, identifying all the genes that expresses as RNA, called the express sequence tags or EST. The second is the sequencing of all set of genome that contained all the coding and the non-coding sequence, called the sequence annotation. So the salient features of the human genome are as follows. The human genome contains 3164.7 million nucleotide bases. The average gene consists of 3000 bases, but sizes vary greatly, with the largest known human gene being dystrophin at 2.4 million bases. Less than 2% of the genome codes for proteins. Repeated sequences make a very large portion of the human genome. Repetitive sequences are stretches of DNA sequences that are repeated many times, sometimes 100 to 1000 times. Chromosome 1 has more genes, 2968, and Y has the fewest, 231. And the scientists have identified 1.4 million locations where the single base DNA differences, SNPs, called the single nucleotide polymorphism that occurs only in the humans. So the Human Genome Project, genetic constitution of an individual lies in a sequence of the DNA nucleotides, as you know. The differences in the two individuals will naturally be reflected in the differences in their own nucleotide sequences. So they can be known only if their entire genome sequence is mapped. Okay. As the young science of genetic engineering developed, the technique to isolate and clone the DNA fragments evolved, and the fast technique of determining DNA sequence was also found out. In 1990, an ambitious project of mapping human genome was launched. The Human Genome Project, or the HGP, was a project of sequencing the entire human genome to find out its characteristic features. As I've told you, the project was estimated to be completed in 13 years. It was coordinated by the US Department of Energy and the NIH, Soon Trust of UK joined the project as a major partner, and in France, Japan, Germany, China was also contributed in this. And the project of the human genome sequencing first phase was completed in the June 26, 2000, and HGP has been called a mega project due to huge cost, estimated to be the $9 billion, a very large number of base pairs to be identified and sequenced. I said about 3 into 10 to the power 9 base pairs, and the large number of scientists, technicians, and supporting staffs were there, and storage of data generated, which requires some 3300 books, which in which each with 1000 pages and each page having 100 type letters, 1000 type letters. So, however, high speed computational devices for the storage, retrieval, and analysis of data made it easier to do the same. But the science of the bioinformatics also developed during this period and helped the AGP. As I have told you, the goals is sequencing, storage, analysis, genes technology, and other methodologies were involved in this. About the express sequence tags, that tags identify all the genes that are expressed in the RNA, and the sequences are the same. And the sequence annotation, the whole genome sequence include both coding and the non-coding regions for sequence. Later, the functions are assigned to the different regions. In AGP, the sequence annotation has been carried out. So the whole DNA was of the cell was isolated, and the DNA was. I'm coming to this. The DNA fingerprinting is a very quick way to compare the DNA sequence of any two individuals. It includes the identifying sequence in some specific region. Okay, and in the DNA sequence as called as repetitive DNA, because in this region a small stretch of DNA is repeated many times. Depending upon the base composition, the length of the segment, and the number of repetitive units, satellite DNA is classified into many categories. The polymorphism in DNA sequence is the basis for genetic mapping of the human genome as well as fingerprinting. The technique of the fingerprinting was initially developed by the Lac Operon, and he used a satellite of DNA as a probe to so high polymorphism was called the variable number of tandem repeats, or the tandem repeat, free and tier. So the DNA Fingerprinting is a laboratory technique used to establish a link between biological evidence and a suspect in a criminal investigation. A DNA sample taken from a crime scene is compared with a DNA sample from a suspect. If the two DNA profiles are a match, then evidence came that suspect. Conversely, if the two DNA profiles do not match, then the evidence cannot have come from the suspect. The DNA fingerprinting is also used to establish the paternity. Here is a very blurred picture as I don't get anyone. The DNA extraction from these cells in a very high speed called the refrigerated centrifuge was done, then amplification by the PCR and the DNA fragmentation by RE, then gel electrophoresis. You maybe not make this technique very well, just know this for now. The double-stranded DNA was split and then southern blotting, then nylon sheet hybridization was done and the nylon membrane passed on the X-ray film and the dark band develops at the probe site. Here is a image. You can refer to this for understanding how the DNA looks like in real life. Okay. In the lab, if you further read about biology or biotechnology or microbiology or life sciences only, you will you will be familiar with this type of things in day-to-day life. So at first the blood sample was taken, and I'll be explaining not only the blood sample, each and everything, how the process is carried out. Here you may refer to this, how the DNA is extracted and DNA is cut by the restriction. Then the DNA fragments are cut. First a sample is extracted, okay? Then the DNA from the blood cells was extracted. Then the DNA is cut into fragments by an enzyme called the restriction endonuclease. Then the fragments are transferred or hybridized to get a multiple fragments called the DNA fragments are separated into the bands called the electrophoresis was done, and then the DNA band pattern on the gel is transferred to a nylon membrane by a process called a southern hybridization, DNA blotting this is called. And the DNA probes binds to the specific DNA sequences on the membrane, then binding occurs. This is known as hybridization, and in this stage the radioactivity probe is bound and the DNA pattern of the membrane was checked, and then X-ray. I have shown you the chart, schematic flow chart only. The X-ray was filmed in placed next to the membrane and to detect the radioactive pattern only. Okay, and from that X-ray film we came to know about the sequence only. This one is okay. This one is this. I hope you understood very clearly as I've explained a lot. So thanks for watching. If you like this video, please share and subscribe among your friends and lab mates. So have a nice day.